TMS protocol in a way that can be feasible and accessible across many different research and clinical environments. The various seTMS studies are ongoing, but have largely been supported by a core pilot grant. So better brain stimulation treatments are needed for mental health disorders. TMS is FDA cleared for use with numerous psychiatric and neurological conditions. Major depression, MDD, is the most common clinical use for TMS and was the first to be cleared for TMS treatment by the FDA. MDD is really a serious condition associated with poor health outcomes, low quality of life, and suicide. And rates of MDD are quite high, estimated to affect approximately 26% of women and 15% of men, but these rates are even higher for military veterans and other at-risk populations. Of patients with MDD, about a third of them do not respond to traditional treatment options. And so for these patients, TMS offers an alternative that can be quite effective for some people, leading to full remission. However, remission rates are less than 40%. So most of these patients remain depressed. This is unacceptable, I think, and likely a result of our limited understanding of how TMS changes the brain in a way that leads to better outcomes. So this is where TMS EEG and TMS EMG come in. We are collectively trying to build a more complete literature on TMS-induced brain changes in relation to things like cortical excitability and plasticity. So I'm an instructor in the Department of Psychiatry and Behavioral Sciences and a research associate with the Department of Veterans Affairs. And in both of these spaces, there is an interest in developing a better knowledge base for the TMS protocols that are used clinically. So as I mentioned, the clinical protocols can be effective for some people, but I think there's really still a lot of room for improvement for all patients. In particular, I think the protocols can be optimized to induce larger and more consistent changes in the brain, more focused changes in specific networks. And also there's a lot of potential for personalizing these protocols for improved brain changes in individual patients. So if you think about it, it doesn't really matter to a patient suffering from major depression whether the protocols work at the group level or in some percentage of patients. So precision medicine takes this perspective that emphasizes adapting care to maximize the benefits for individuals. So for TMS, this might look like choosing personalized brain targets that can be stimulated to treat the patient's specific set of symptoms. Or this could look like using a patient's own neurophysiology in real time to adapt how we're applying the TMS to increase how much their brain is responding to it. These are just sort of two examples of what precision medicine with TMS might look like. But in order to take any precision medicine TMS approach, a lot more knowledge is needed about how the TMS interacts with neurophysiology and behavior. So I use neurophysiology to try to understand changes in the brain over time, including with the TMS protocols. So my research really covers 5 focus areas. These include brain stimulation interventions, novel methods for brain stimulation, sensory motor interventions, cognitive and motor rehabilitation, and I study theories of predictive neural dynamics. So with TMS EEG, this is largely within these first 2 areas. So I study sensory EEG confounds in TMS EEG studies, and I've developed methods to minimize these confounds. I'm working with TMS EEG methods for predicting post-surgery delirium and understanding different types of dementia. I'm not going to get into that work today, but please ask if you'd like to know more. And I study the neural effects of TMS in order to develop novel protocols. So in this work, we think a lot about something called closed-loop TMS EEG. So this means using EEG in near real time to trigger the timing of TMS pulses. This can increase the effects of TMS on the brain. We also think a lot about computational and experimental parameter search techniques to figure out the best sort of combination of settings for the TMS that can optimize TMS effects on the brain. And we use context to put the brain in a defined state that may be most sensitive to the TMS. So this is really what we'll be spending the most time on today, because sensory entrained TMS is a type of TMS protocol that uses musical rhythms to maximize excitability of certain networks at very specific times, and then applies TMS at those times. So seTMS is synchronized music with TMS that increases the effects that the TMS has on these brain networks. I'll show you exactly what this means with some data, but first I want to show you the tools that we're using and describe the kind of data that we'll be looking at. So we are using the CORIT TMS EEG facility. We use the TMS device as well as the TMS compatible EEG system. We also use Neuro navigation. So this uses motion capture technology to track the participant and the equipment in the room in relation to the subject's MRI. The Neuro navigation software shown in this photo is Localite, but we have converted to another software that Chris Klein is developing that's more flexible. Chris's preprint on NaviNibs is now available. If you'd like to learn more, I encourage you to look it up. NaviNibs is a free and open source neuro navigation software. We also use the cobot, that's the robotic arm in the room. The robot holds the coil to the participant's head and moves with them when they move to keep the stimulation on the correct target and also to quickly adjust TMS coil placement. So for those of you who may not be familiar with the cobot, this is a video of the cobot placing the coil on a colleague's head. It's pretty cool. We're lucky to have access to a couple of these robots. Not many labs have access to these because they're expensive and they can be very clunky to use still. But they are allowing us to stimulate in ways that are not possible when a person has to manually hold the coil to the head. That's pretty common. I have considered these to be sort of something that won't become too commonplace, but I'm starting to think differently about that. One of our research assistants found a robot that looks almost exactly the same making espresso drinks in a coffee shop. So I think they may become more accessible research spaces too. Here's another photo. This shows, I think, more clearly the right hand. So we attach surface electrodes to the skin above a muscle in the hand to measure muscle activity when we stimulate the primary motor cortex. So when you apply a pulse of TMS to the motor cortex, you can stimulate the full corticospinal pathway and evoke a response in the muscle associated with that area of M1. So this muscle response is called the motor evoked potential, or MEP for short. Here's a schematic of the setup. So TMS is applied to the scalp. This evokes an MEP in the hand when stimulating M1, but we can also evoke, a response in the EEG called a TMS evoked potential. So the TEP is important for understanding TMS effects on the brain, especially when you're stimulating outside of motor cortex, where we wouldn't get an MEP. So clinical TMS is most often applied to the dorsolateral prefrontal cortex, DLPFC. So one thing that we work with is the TEP from DLPFC stimulation. So today we'll be looking at sort of 3 different types of data. One is EEG recorded during listening to rhythmic sounds. So this is without TMS. We'll also be looking at MEPs from stimulation of the primary motor cortex, and we'll be looking at TEPs from stimulation of DLPFC.
Can I ask a question?
Yeah.
Hi, it's Tony Archer here.
Hi.
So the TMS evoke potential, so this is not only an evoked response from the target region, is that correct?
I'm sorry, I'm having a little bit of trouble hearing, but yeah.
This is an evoked response from the target region, right?
Yeah, that's right. So if we apply the TMS to DLPFC, then we can record the TEP from the electrodes under the coil, but also in other areas of the brain too. So sometimes, for instance, we'll stimulate DLPFC and then look at a response in like parietal cortex or something. But yeah, what we'll be looking at today is DLPFC.
Okay, so it's the evoked response generated in the DLPFC, but then it would propagate to other places, presumably, and then you could record the downstream effects from
Exactly.
DLPFC stimulation in other brain areas.
That's right.
Okay, so then how do you select out the TEP from DLPFC? Is that just based on using electrodes, or is it source localized?
Okay, this is a really great question. We do both of those things. Without getting too sidetracked, I'll also say that this TEP response sort of reflects more than just the local cortical response. There's sort of other things that sneak in here. There's sensory responses and stuff too. But we do look in the sensor space and also in source space. But there's really a lot that we're still trying to learn about these TEPs. Does that answer your question?
Thanks.
I also want to point out that we are using MEPs and TEPs to develop these methods and to try to understand the neural effects of these methods. But what is realistic in clinics for TMS treatments is more limited. So we have all this fancy equipment, and that's really helping us to learn about TMS. But the only realistic equipment for administering treatments often is the TMS device itself. So most clinics don't use MRI guided neuronavigation even, let alone EEG or EMG or robots. So I think that's really important to keep in mind here too. So one thing we've learned from this TMS EEG and TMS EMG work is that there may be optimal brain states for inducing the largest changes in the brain with TMS. One approach for taking advantage of these brain states is to use EEG to trigger the timing of TMS based on predicting when these states are going to occur. This type of closed loop TMS EEG, I think, is exciting scientifically, but impractical in many clinical settings. However, we can induce brain states for TMS using sort of other methods. And this is a novel approach based on my work tracking EEG dynamics during sensory event processing. And so we're going to dive into that research a little bit next. Predictable sensory events can align the phase of certain types of brain oscillations. So this is my work in this area is sort of based on this idea of covert motor activity. So generally speaking, brain systems supporting body movement are active when we're listening to music passively without moving our bodies. This covert motor activity is not really well understood, but some theories propose a role in auditory timing prediction facilitated by motor timing. So one question is how music-related covert motor activity is related to motor activity during overt movement. So we tried to address this question using EEG by measuring mu rhythms. So mu in EEG is recorded over sensory motor cortex and shows a characteristic shape in the time series data that results in a spectrum with a fundamental in the alpha band and also another peak in the beta band. So lateralized mu enhancement over hand areas during or just before foot movement is thought to reflect hand movement inhibition during movement activity of another effector. So we recorded EEG during silence without movement, during overt movement of the hand or of the foot, and also, excuse me, during music listening without movement. We used an ICA-based dipole clustering technique and identified 3 mu-related clusters. One that localized to left primary motor cortex, one that localized to right premotor cortex, and one to like midline premotor cortex. Right foot tapping was accompanied by mu enhancement in the left lateral source cluster. This sort of replicates previous work that suggested that this reflects hand movement inhibition. We also found that music listening was accompanied by similar mu enhancement in the left as well as midline clusters. So we were the first to report and also source resolve music-related mu modulation in the absence of overt movements. So this covert music-related motor activity has been shown to play a role in beat perception, and these results show enhancement in somatotopically organized mu, supporting overt motor inhibition during beat perception. So then we...
Can I interject something here?
Definitely.
I don't want to distract it too much, but there are also beat-related responses in the evoker.
Yes, that is a great question. I'm going to get into that actually a little bit.
Okay.
Yeah, but that's absolutely true. So we then sort of proceeded to studying the dynamics of mu and other oscillations over time around sensory events, the various signatures of these changes in EEG, and also what factors can be used to manipulate these effects, as well as other sensory motor and perceptual measures. So some of this research was motivated by the work in MEG and EEG beta band activity during music listening. This has been carefully studied for the last 15 to 20 years by people including Takako Fujioka here at Stanford, and also John Everson at UC San Diego, and now also McMaster University in Canada. So these studies indicate that there are beta band modulations that are involved in predicting the onset of auditory rhythmic events, and can be the result of top-down influences of perception of the beat. So in our subsequent studies, we sought to better understand the sensory motor implications. We used EEG. So one thing we asked was whether similar processes are recruited during visual versus auditory rhythms. And we used a beat omission paradigm. So this sort of speaks to your question. So we were able to observe separately the signatures of predictive timing and stimulus-driven activity. And we find some modality-independent markers of rhythm prediction. We source resolved the activity, which revealed that there may be overlapping networks of predictive beta activity in parietal and frontal regions, auditory specific predictive beta in bilateral sensory motor regions, and visually specific predictive beta in midline central and bilateral temporal parietal regions. Although we focus broadly on beta band activity in this paper, this included beta activity from sensory motor mu dynamics. Okay, so what I want you to take away from this work is that we can track phase dynamics of oscillatory brain activity when we're listening to sounds that have predictable and engaging rhythms, or in short, that sounds with predictable rhythms create fluctuations in brain excitability with consistent timings around predictable auditory events. We show that these EEG dynamics occur in frequencies that should be relevant to TMS brain excitability and in networks that are known to be involved with predicting the timing of sensory events, such as motor planning networks, the dorsal auditory stream, and also the salience network. So mu is what closed loop TMS EEG targets because it's thought to represent cortical excitability. Mu synchronizations and desynchronizations can be studied using the power spectrum or by looking at intertrial coherence, ITC, of the time frequency activity. So if you look at the ITC time series, the times of highest excitability should be reflected by ITC troughs in mu. So we're finding that the timing of these ITC troughs in mu all appear to be pretty consistent around 200 milliseconds prior to predicted musical events, regardless of the tempo of the sound. So then,
Oh,
do we have,
Tony's got another.
yeah, I was just
Yes.
gonna ask if you looked at evidence of cross-frequency coupling. So the prediction would be if mu is being modulated by the beat, its envelope should depend on the beat frequency. And that would show up in a cross-by spectrum analysis, which we could talk about later. I'm just curious whether anybody's looked for that evidence of coupling from the beat frequency to the envelope of the mu rhythm.
actually, yes, that's a great question. There is some work on that. So, Sylvie Nazardon does
Yeah.
something like this, where she looks at specific frequencies that are present in the musical rhythms and finds signatures of them in the spectrum. This is a little bit different because I think we're looking more at sort of evidence for synchronization and desynchronization around the beat. And we're more interested specifically in the timing of activity before the beat, because we're looking at this sort of like timing prediction, using the motor
Yeah, I
system.
think with Sophie's work and others like Blair here at Stanford, these would be the signal that would trigger that. So the beat has to be detected somewhere in the brain first before it can modulate mu. So
Yeah, absolutely. Yeah.
that there's a whole other system that you could be targeting, which would be the trigger for the mu modulation.
Yeah, that's a really good point. I mean, we could be sort of targeting different responses to the sounds. Absolutely. I think for corticomotor excitability, this approach sort of makes sense to me, but yeah, that's a really good point.
Can I just chime in here also?
Yeah.
Jess, hi, Dan Bowling. But so the presumption is that this modulation of mu is happening. You said it's tempo independent, but you're just looking at it tempo independently, right? I mean, it is tracking the tempo of music, presumably, right?
Okay, so of the different tempos that we have looked at, we consistently find that it's about 200 milliseconds, maybe a little bit less, prior to the predicted beat time.
Right.
Okay. But yeah, there could be some variability in that, I think.
I got you. But that's happening on the cycle of the beat. You're getting a envelope.
As long as it's a predicted beat, then it's about 200 milliseconds before.
Right.
Yeah.
Cool.
I know. It's cool. So, okay, so then this is where Corey and I come in. We recruited a team to work with us to develop an approach to TMS that induces these mu dynamics and then times TMS appropriately to maximize the effects of TMS. So we've started with TMS to the primary motor cortex and are also interested in the prefrontal cortex. So here's a single subject example of EEG during listening to a musical sounding metronome. So on the left is the ITC of the event-related spectral perturbations from EEG electrodes over motor cortex. In contrast to the previous papers, this has been averaged over all trials. So this vertical dotted line that says 0 milliseconds, this is the time of all the beat events. Below are the average alpha and beta band ITC traces, showing an alpha ITC trough around 200 milliseconds prior to the beat. And on the right is, excuse me, the same but recorded in electrodes over prefrontal cortex. So one thing to note is that this EEG is at the sensor level rather than in source space. So the beta band activity in these data have mixed sources rather than more isolated mu-related activity. I think this is why we see sort of a different pattern here in alpha and beta, and then in the prior papers. I think it's to be expected, actually. And this increase in beta ITC with simultaneous decrease in alpha ITC, I think represents a fluctuation in motor cortical excitability. So for motor cortex and for prefrontal cortex, maximal excitability, I think should occur about 200 milliseconds prior to musical beat events, because this is when we find the network-specific and frequency-specific ITC troughs. So TMS pulses and seTMS are applied at this time in relation to musical beats. So this is why closed-loop TMS EEG is not really needed with this approach, because if we know that these brain states occur 200 milliseconds prior to each rhythmically predictable musical event, then we can use the sounds alone to trigger the TMS pulses. So in practice, we won't need the EEG.
So what are you targeting?
What part of the
What's
brain?
target? What, yeah,
So we started with primary motor cortex, and then we're sort of moving into DLPFC.
You said musically sounding metronome. Can you just, what is that? That's like a
Yeah,
kick drum or something?
nice catch. Yeah, so we do music for some of our protocols, like the plasticity protocols I'll tell you about in a little bit. But for these single pulse paradigms, I've been using a like a metronome sound that has alternating strong and weak beats. So it sounds sort of musical. It's also what we used in our prior EEG dynamics study. This is sort of to reduce the number of things that are happening between the beats so that we can, it's a little bit easier to interpret the EEG signal between the beats rather than using complex music with a lot of things going on.
Like a TikTok situation.
TikTok, yeah.
Cool. Not the social media platform.
Oh, that. Oh, yeah, not the social media platform.
What was the speed of the tempo in that
So
experience?
we have tried a few different ones, but what I'm showing you today, the strong beats are once a second. So the weak beats are between those.
so something I think that is promising here is that you get the generalization to prefrontal. So your theory is that the synchronization is really a motor kind of phenomenon. You're like tapping to the beat implicitly. And if it were just a motor phenomenon, that synchronization would be restricted to the motor system and maybe the auditory, primary auditory system. But you wouldn't expect it to generalize prefrontal cortex in a simplistic model. But it seems like it does go to, you get a beat related ITC signal in your prefrontal electrodes. The fair assessment?
Yeah, that's a fair assessment. There's a few caveats. I think one is that, you know, we could be picking up some signal from other sources in the brain. This is why it's really important that we also move to source space and really try to understand these better. However, we did already sort of do this in our prior studies where we were looking at sort of different networks that may be involved during the visual and auditory metronomes. And in that, we did find some indication for overlapping or visual and auditory beta, predictive beta in frontal areas too. I think there's a lot more that we need to understand about that. But yeah, your assessment is fair.
So for your surface analysis, are you just looking at topographies and time frequency spectrum from average reference data?
Are we average referencing? Yes.
Yeah. So what you could do very quickly to restrict the locality of the surface data is to do surface line Laplacian.
Oh, sure. Yeah.
And
Okay.
that would eliminate some of the kind of volume conduction confounds.
Okay, that's a
And
great
that's
idea.
really super easy to do. And if it survives that, if your prefrontal signal survives that, then it's, you know, even more promising that it's actually generalized to your prefrontal target.
Okay, that's a great suggestion. I think we have started using those methods a little bit for other projects in the lab, and so I think, yeah, this is absolutely something I can do. Thanks.
I'm curious about how replicable this time blocking event, this response to music within this subject, like across different time, across different days, across trial.
Test retest of this. Yeah, it's a great question. So we are sort of looking at a few different things. I can get into intersubject variability a little bit more if you'd like, but also across time. You mentioned like if we do this and then they come in and do it again. We do have a few subjects who have been in multiple times, and we're starting to look at this in visit 1 versus visit 2, and some people in visit 3. And it seems fairly consistent so far, but we haven't done sort of a larger scale study on test retest of this. But I think it's a really important question. We also have some test retest within session in the plasticity study. I'll get to that study in a little bit, but basically we make these listening recordings at multiple time points in the session so we can kind of look to see if they change. And we're seeing a lot of consistency between them, but that's just sort of my general assessment from what we have so far.
Is this calculated by event related spectral perturpation, the kind of like ERSP, by removing the baseline period? Or normalizing using the baseline period.
So we use a baseline sort of before this period, but is that what you're asking if we baselined?
Yeah, like removing the baseline activity in the event activity, but by just a normalization equation.
I think I'm missing the question.
Okay, never mind.
Oh, I'm
Yeah.
sorry. Can you say it again, maybe?
I'm thinking about this time frequency map, how it's calculated. Is it from is it by removing normalizing using the baseline period of activity?
Yes. Okay. Yes. So baselining gets a little bit complicated since we're looking at this sort of pre-beat period. So we use a baseline that happens before this. I think I have to look, but I think it goes up to like negative 250 milliseconds or something like that. But yes, we use a baseline period.
I guess that's not really a baseline because there was a sound 500 milliseconds before, right?
Right.
Or is that
So
it?
if you have a sound and then yeah, and then 250 milliseconds and then 250 milliseconds.
Okay.
The
Do you see?
selection of baseline period also influences the result.
Yeah.
Yeah. Because how to define which period is more baseline? Because before the time 0, most period can be like baseline.
Before 0. Yeah, but that's not really our baseline because that's our window of interest.
Yeah, that's true.
So this is unnormalized, right? This time frequency spectrum. It's just the time frequency spectrum. Is that correct?
Is it normalized? So these
or not?
are not, no.
Yeah, yeah.
Oh,
So,
okay.
so it's,
I thought
yeah.
it was-
So one comment, if you look at the beta band in the prefrontal coherence for music panel, you can see the beat modulation. There are two, there's like a red thing, blue thing, red thing, blue thing, and that's the frequency. Or I'm asking, maybe I should ask, is that the frequency, is that tied to, is that modulation tied to the frequency of the beat? It's clearly tied to milliseconds from the beat, but is it must be tied to the beat frequency?
Is this, are you asking if this is time locked
Yeah,
to the beat?
to the beat. So is the envelope of beta time locked to the beat frequency?
So, yeah, so this question, if I'm understanding your question correctly, you're asking if this is time locked to the 0 milliseconds, which is a little tricky to look at here. It's one thing we've done with the ERSP to try to understand timing is by looking at the slope from the minimum to the maximum in the spectrum, because you know, sort of the timing and the number of times that are affected by this type of analysis can change based on the parameters of the analysis. But we do sort of look at so that's one thing we do is we look at sort of minimum to maximum and slope. Does that answer your question?
Yeah, I wasn't quite sure I followed, but why don't you keep going? Let's just let aside,
Okay.
side observation.
Okay. Okay. Sorry, I feel like I didn't answer your question. But okay, so this sensory entrained approach to TMS, because EEG may not be needed, it's easy to implement on a budget and in different environments with minimal equipment, minimal setup time, no extra training in EEG necessarily. It really requires headphones and then a software that can trigger the timing of the TMS pulses. So we've developed single pulse TMS protocols and 2 repetitive TMS protocols. This schematic is of single pulse seTMS. So the participant hears through earbuds. We also use over-the-ear headphones. And the TMS pulses are applied 200 milliseconds before some of the music events. In our first study, these pulses were applied more than 3 seconds apart. So this allowed us to look at MEPs and TEPs after each pulse. And also because the spacing isn't thought to induce plasticity. So let's look back at this subject. This is the same person we looked at before. We know this person had alpha ITC troughs about 200 milliseconds prior to the beats. So in this subject, we also recorded MEPs during single pulse seTMS and standard single pulse TMS to motor cortex. And we recorded TEPs when these protocols were applied to DLPFC. We found in this subject that MEPs and TEPs were larger in the seTMS condition than in the standard single pulse condition, indicating potentially larger stimulation effects. After a few pilot subjects like this, we decided to proceed with the first study that focused on single pulse protocols to M1. So here's 27 participants with the average motor ITC time series in panel B. So the alpha band ITC trough in this group is present at the group level as well as in individual subjects. When we look at the average MEP sizes for seTMS here in red and standard TMS shown in black, we also see that the MEP size is larger, and this happens very consistently across participants. So here we have 19 participants. We also, you may notice there's a third condition here in gray. We have some control conditions designed to test whether this effect might be explained just by the presence of music, rather than the timing of TMS with low ITC, mu ITC. The control shown here used the same sound as in the seTMS condition, but the TMS was applied at a different timing. So far, using the control timings, we find that the presence of music is not what is driving this effect, but rather the timing of the TMS. When we,
So when is the TMS pulse?
for the control, so we have
No,
like,
no, in both. In both I guess in the red and the gray, when is the pulse?
okay, yeah, so in the red and the seTMS condition, we applied the pulse 200 milliseconds before some of the musical events. And then in the control condition shown here, the pulse was applied at 0 milliseconds, so at the time of the musical event. We also have a plus 20 milliseconds condition where the pulse is applied 20 milliseconds after the beat.
And that 200 milliseconds is like supposed to be aligned with the alpha trough, or what was why have you picked 200 milliseconds? Is that why you picked 200 milliseconds?
Yeah, that's right. So this
Okay.
is, yeah, based on sort of the group level.
Maybe that alpha, like decreased alpha would be like increased excitability.
Yeah, right.
Okay.
And the beta peak as well, right? Like you're getting some time locking of things happening 200 milliseconds before the beat. That's why you picked it.
Yeah, so we're kind of focusing a little bit on that mu trough. That was what we were originally sort of interested in. But you're right, there is this synchronization of beta at the same time. And so I think this does indicate a change in excitability.
I guess one question, is it always the same? And if there's participants where they're different, if you picked the time that was the beta peak or the alpha trough, like would you see something different? I don't know. I mean, that's a big experiment, but.
No, we would like to do that. So we'd like to personalize sort of on lots of different measures. We started with this sort of group level approach, also because it's if the idea is to provide a technique that is easy to use without EEG, we want to see if it works sort of with this generalized timing. But I think it is really important to look at, if you apply the TMS pulse at that person's sort of individualized trough time, will work better. We are interested in this question.
Okay.
And also if different types of sounds might work better than others or different, factors, but yeah.
Well, yeah, looks great. Very
We
cool.
we also want to do an experiment that has sort of like more controlled timings, sort of a range of timings,
Yeah.
planning that too.
That would be cool.
Yeah. So we just have some pilot data with the TEP from DLPFC stimulation. We're finding in our first 8 participants that the TEP does appear to be larger with single pulse seTMS than with standard single pulse TMS. I think one thing to think about is that when averaging waveforms, many things contribute to the amplitude of the average waveform, including variability in timing and in amplitudes, right? So we might find, for instance, that this larger amplitude TEP may be related more to more consistency between TEPs when using seTMS than when using standard, but really we can't make any actual conclusions like this until I've analyzed more of the data. But we're wrapping up data collection next week for N equals 25, and I'm going to dive a little bit more in depth to these TP results. We also have data using one of our repetitive TMS protocols, seITBS. So this protocol is identical in most ways to the clinically used intermittent theta burst protocol. So ITBS applies 10 50 Hertz triplets of TMS pulses, which is repeated 20 times in sequence with 8-second gaps between each repetition for a total duration of 3 minutes and 20 seconds. So seTMS, the seTMS version of this, plays the music in such a way that these pulse triplets land in ITC trough times. Because the triplets of the protocol are timed with maximal excitability states, this plasticity protocol should be more effective. than standard ITBS. Plasticity from ITBS usually lasts between 30 and 60 minutes. And plasticity in this context is assessed by looking at change in the size of the MEP when applied to M1. So here's one participant's MEP data from before and at multiple time points after standard ITBS in black and SE-ITBS in red. So the y-axis is MEP size, the x-axis sort of follows the progression of the experiment. So for this participant, standard ITBS was first, followed by a break to let the effects wear off, and then SE-ITBS. But the order of protocols is being randomized across participants. The vertical dark gray bars here and here, these indicate the times that the TBS protocols were applied to M1. And again, plasticity is when these MEP sizes after the protocol are larger than the MEP before the protocol, MEPs before the protocol. So you can see here that plasticity was induced in this subject with both these protocols and was larger with SE-ITBS than with standard. We have 20 participants with these two protocols applied to M1. So the average across participants is shown here on the left in panel A, indicating that at the group level, we're inducing plasticity and that this plasticity is larger with SE-ITBS than with standard. On the right, I'm showing all individual participants. You may notice there's a lot of variability here in the time course of the effect and how much plasticity. This is quite common. There's a lot of variability in ITBS effects, but in most everyone so far, we've induced plasticity that's in general greater with SE-ITBS than with standard.
This is all motor, right? This
is all motor. Yeah. Yeah, we are sort of moving into DLPFC, but I think a lot of the sort of foundational work with seTMS and trying to understand these effects can be done first with M1. Should be done first with M1.
I just ask, when you're looking at individual variability, either in the plasticity protocols or just in the time frequency analysis you were doing, does it relate to musical experience or
I'm so glad that you asked this question. So we are looking at this. We are interested in this. One of our research assistants, Lily Foreman, she's on the call. She's been doing a lot of these analyses. Actually, I have. Oh my gosh. Okay. So we do collect some information about musical experience, also dance experience and other hobbies. What we're finding, so in panel A here, this is just like showing the timing of the ITC trough in musicians and non-musicians. We don't actually see an obvious difference here. And then also in the MEP effects, we don't see an obvious difference here. This was surprising to us a little bit because there is some work. Michael Hove did some TMS work with single pulse TMS on the beat for different types of musical sounds. And he did find some interesting differences between musicians and non-musicians. But there's really a lot of variability when we look in the literature that would indicate that this may or may not happen. We recently got a C# voucher to study individual differences in both the ITC dynamics, but also the seTMS effects. This is with Takako Fujioka in the music department. So she's been studying these types of dynamics for a long time, as I mentioned, and we sort of share this interest in individual variability. This grant, the idea is to use a finger tapping paradigm to see if we can categorize people not by their musical experience, but by their ability to synchronize with the beat. So she's developing this at-home finger tapping task, and then we're going to be doing seTMS. So this is something we're interested in. I'm not sure that the musician, non-musician split, I think that might be a little bit too non-specific to separate out these effects, but there may be other sort of ways that we can think about individual differences and personalization.
Like dancer versus non-dancer.
Yeah, that's an interesting one.
Yeah.
It's really hard to define, right? Like if somebody's
Yeah.
a dancer, or if you can be super musical and not a musician, or not consider yourself musicians.
Yeah, and how
I
you
don't-
do define musician, like we just said, 1 or more years of musical training or experience, but like, it's hard to sort of categorize. I mean, how long you've been playing and what you've been playing and what kind of music and what is your relationship with rhythm, and there's so many different things to consider.
Do you think that if you like added more modalities, like sensory modalities, you would like get like a dose dependent increase in the effect? If you had like a visual stimulus that was also entrained, or like a somatosensory stimulus that was also entrained at the same beat, do you think it would get bigger and bigger, the effect?
I really love this question. Yeah, I don't know. I mean, it does seem like in some networks, maybe using a visual metronome or a visual stimulus would work better. And then what happens when we sort of combine modalities? Would we find a more robust effect? It's a great question. We could
Also
have
kind
this
of
whole
relating-
multi-sensory TMS experience,
Yeah, virtually.
which would be
Just-
super fun.
Disco team, yeah.
Also, like relating to Tony's comments about how this trough that you're looking at propagates into the area that you're interested in stimulating for depression. I guess having lots of like multisensory inputs might engage more networks and therefore lend this to more TMS type.
Oh, yeah,
Is-
that's
is
really
it-
great point.
Is the music during like the ITBS, is it the same like kind of TikTok sound, or is it a little bit more complex, or?
No, actually, in the ITBS protocol, I forgot to mention, I think that we're using music during ITBS. So just regular single pulse, and then during the ITBS protocol, we have a few different musical sounds we use, but they all have the same tempo as the metronome. But yeah,
Okay, what kind of music is it, or?
it
Yeah.
is high groove music. So there's this whole sort of area of research, music cognition research, that looks at something called groove in music. And there's a very convenient for us library of musical sounds, musical clips that have been rated for their level of groove. Groove just means the amount of sensory motor engagement. So sort of a lot of different factors have been studied with regard to groove, but essentially what has been found is that there's some music that we are better at synchronizing with, and this is also more enjoyable to us. And so anyway, we chose 3 musical sounds from the groove library that have been consistently rated very high in groove, and are sort of within our tempo range.
Do you adjust the tempo a little bit?
Yeah, we do.
Okay, to match the ITBS?
Just slightly, yeah.
And is the ITBS speed that you're using the standard ITBS, like gamma or whatever, like, is it the same normal theta burst speed?
It's the
50
same,
hertz?
yeah, yeah.
50 hertz, okay.
It's the same. We've really spent a lot of time trying to figure out how to sort of keep the same protocol. That
Yeah.
way we're not manipulating the clinical protocols.
Yeah, that makes sense, yeah.
Are you time stretching to adjust the tempo? Like down to, because you say are you trying to bring it down to 60 BPM from say 98? Or
Yeah, 120 or 60, basically, yeah. So we've chosen the stimuli that we can sort of, from the group library, that we can sort of best use, but yeah. It's not great. We need more that are just like 120 beats per minute, I think.
But is the downbeat of the music, is it timed to be 200 milliseconds before like the
Okay,
data burst
yes,
starts?
it's- It is consistently 200 milliseconds before. That's our timing, before the actual
The downbeat, okay.
But
Oh, yeah, that's right, yeah.
if you take Baby It's You and you want to put it at 60 BPM, I mean, it's already a multiple of that. right, at 120.
So that, yeah, that one's easy.
But you could, I mean, you're gonna cut the tempo in half, right? So it's, is that what you're doing? Like
No.
you're taking the, okay.
I guess they mentioned 120, I'm guessing for all three of those. So they shifted 120 beats per
I
minute,
see.
I'm assuming.
So you're bumping out of space up to 120, essentially.
Or is
Right.
that right? Or would it be 100 beats per minute? No,
We can,
but yeah,
we can,
120.
doesn't matter.
Yeah, the question was about whether you're manipulating the actual BPM to better fit your
So
protocol.
in these, we didn't, but we have talked about that in some of our grant proposals because to use sort of a wider variety of music, I think we're going to have to consider either composing our own music more likely or sort of manipulating the music. I don't want to manipulate the music too much though, because it could change people's experience of the music and their sort of sensory motor prediction of the beat, I think, a little bit. So
maybe just to clarify, what you're doing is you're timing your ITMS pulses before the downbeats in these songs at these tempos. So that's what you're shifting. You're shifting the stimulation relative to the beat, so you don't need to.
So you don't want to change the tempo.
we have the timing of the beats already. So, but yes, that's right. So we're playing the music in such a way that the stimulations will be correctly timed to the beat.
Do you like, are there gaps? Do you pause the music or something, or and start it, stop it and start it or something like that? Or maybe I'm not thinking about this right.
Do we stop the music?
So the music is playing for like 5 minutes while you're doing the ITBS for 5 minutes?
It's playing for 30 seconds on a loop. So it's the same
Okay.
30 seconds.
Okay.
I know it's probably a little bit annoying, but
Yeah.
yeah.
Okay. Well,
Because
see,
30
I wanna be a-
seconds, I'll tell you why, because the
I'll
30
be a
second
participant.
clips are so the whole groove library was sort of developed using the free iTunes previews, which are 30 seconds long. So we're using those same 30-second clips.
Okay.
So can I just bring you back to this, do you have more to go? Maybe we should let you finish.
No, please. No, not
Okay.
really. No.
So this issue about the DLPFC stimulation, right? So it makes so much sense from a clinical perspective, and that's why you want to go there, obviously. But this notion that the timing is the same, I'm
I
just trying to
know.
wrap my head around that, right? Like that there's I mean, the models that we think about in terms of auditory motor entrainment, are specific to, auditory communication with motor and premotor areas. So the notion that you're picking up in the DLPFC at the same lag.
Yeah.
I mean, what does that imply? Or do you think, do you really have to wait for the source separation to see whether or not, or I'm playing catch up with what you're saying, do you do an analysis where then you can segregate and differentiate these responses? But what do you think?
This was surprising to me, actually. I expected that different frequencies would be relevant and different timings from the beat. There's sort of an enormous amount of literature on gamma band, alpha band, and beta band oscillations around the beat. But of course, we looked in alpha first because we were already doing that with motor cortex, and this is what we found. So I do think we need to sort of dive into this a little bit more and see if there's sort of different parameters that are needed for prefrontal cortex. But so far, this is what we find. I'm not sure exactly how to interpret it because mu doesn't really localize to dorsolateral prefrontal cortex. So I'm not sure. Yeah, it's a great question.
So like the null hypothesis would be that you're getting bleed over from just like.
That would be the null hypothesis, yes. Yeah.
Have you tried any 1 Hertz like TMS protocols with this? Because I know you were saying that you use 60 BPM, so it kind of lines up very well, and that's what we do in our labs. So.
Yeah. So, developing these protocols takes a lot of sort of mental gymnastics, but we have sort of worked out how to do a 1 hertz or 10 hertz protocol actually. But we haven't used it yet in any subjects. We sort of started with ITBS. But yeah,
Yeah,
we
1
could
hertz-
do 1
1 hertz would
hertz
be-
easily.
1 hertz would be really interesting.
Yeah, I think that would also, like, for us in particular, actually, when we're doing, so I do TMS EEG, I work in the lab that does it with kids with epilepsy, and our goal is to see inhibition in sort of the motor areas that you're talking about, that you're already measuring.
Oh, interesting.
So, I think we should probably talk, I'm not
Yeah.
I'm just like a research associate, but like, I'm sure that the doc would be very interested to hear from you, because if we could get more of a, like, an inhibition as a result of the music, it would be awesome. And then also the kids too, I mean, when they're sitting in the chair, they're very, they don't like being in the chair, they don't like moving around, they like to move around a lot of stuff, so if they had music going at the same time, I think that would also be helpful, as like a doubly beneficial thing.
Yeah, there's a lot of points in there. So I do think that sort of thinking about brain stimulation in this way, as this multisensory kind of experience, could be valuable for other TMS research and in other clinical populations, and that's a really good idea. It's a really good point about the kids. And then also, yeah, for anyone who's been a subject in a TMS experiment, they can be tedious, and especially so for children who have to sit
Yeah.
for a long period of time. And one thing that some people have done, at least in the clinic, is to have like, and probably in research spaces too, to keep the participants engaged, is to like have a video playing or have music playing at the same time. And this seems like a huge experimental confound to me, but if you use the music or the video in such a way that it actually supports the brain stimulation, then it could be also, you know, it could help in a lot of ways, keeping the subject awake, alert, interested, and then maybe also boost these TMS effects.
Yeah,
Yeah,
we, to avoid confounds, we don't, like, the kids aren't watching or listening to music or anything when the, like, 1 hertz is happening, and it's like 17 minutes long, so a lot of times it's very, I don't know, it's tough on them, they're not interested in doing that, they don't like it very much, so if we could have music at the same time, and it was somehow also benefiting the plasticity, that'd be pretty cool. So,
I think all around it could be more fun for everyone.
yeah, I mean, it's not gonna be fun either way, but more fun.
I want to just add in this context that, you know, the time stretching algorithms have gotten so good now. And so some of the work that I'm doing in my lab is like to develop, you know, web-based time stretching. So you can take any song, you can ask them what their favorite track is. You can time stretch it down to, say, 60 BPM or 120, so you don't have to adjust your stimulation protocol. And then they're getting the benefit of listening to their preferred music. And you can just, you know, you can use music information retrieval to get the downbeat and then, you know, deliver in this way. I mean,
Yeah,
I think it really, in
That
terms
sounds-
of improving the experience for the patient.
that sounds great. I think there's sort of a lot of issues that come up when we're using actual music. I'm curious to hear your thoughts on. So one of our collaborators, Scott McCaig, has been sort of encouraging me for years, and I've been ignoring him, to compose our own music. It sort of
Yeah,
bypasses all the sort of legal issues, but also then you can compose music that engages mu dynamics the most and at very specific times, and you can manipulate things like tempo a little bit more. I don't think I'm the right person to compose these sounds, but
Generative AI.
yeah, there's
Yeah.
a website.
Yep,
But you
yep.
know,
have
know,
AI
it's-
do it. But then, like, yeah, sure.
Generating knockoff Taylor
Is it going to
Swift.
sound really electronic, or is it?
the music's already so good at this, right? Like, this is what music does, right? Like, this is, I mean, what people have been doing for so long is creating music that's going to entrain your brain and do it effectively at certain
Yeah.
frequencies. So all you have to do is just tweak it. You just, I mean, the legal stuff is a disaster. But you know, in an experiment, right? You can just shift it to exactly what you want. Like, those rhythms are in
sure.
there, right? So then you can
Okay,
just bump it.
I like
to-
the idea of.
I mean, we
using it.
should probably do that and also collect, like some data on their perception of the music and how much it makes them want to dance or something and how much they like it. But yeah, I
Right, go-
think it's a good idea.
So we're actually 10 minutes over. Everyone's clearly very excited about your work. I think we should let everyone go, but thank you so much and congrats on your C# voucher.
Thank you. Thanks, everyone.
Thank you, Jess.
Cool, yeah.